Good evening, everyone. Today we'll start DNA sequencing class. Today we have some practical class. Okay, give some how to do this sequencing. I will show in practical way. Yesterday, any doubts from yesterday's class? So many are absent in yesterday. Today also maybe I expect the same. Any doubts from yesterday's class?
You can do several steps.
Yeah, okay. Yeah. What is sequencing? What is DNA? What is sequencing? It should be sequencing. It's not DNA sequencing. What is sequencing? Nucleic acid sequencing. What is sequencing of DNA or RNA? Either it can do RNA sequencing also. What is the meaning of sequencing? Can anyone tell?
So determining the order of nucleotides in the nucleic acid.
Just deducing what is there. Okay, just ATGC. Which base is there and how it is one after the other, which base is present. It's not only order, we are knowing what is there in this position. Okay, we are determining actual basis of the DNA and its order. Okay, it is called as sequencing. How we can do that today we are going to discuss. Sequencing, either it can sequence DNA, either it can sequence RNA, either you can sequence protein. Which sequencing came first actually? Which method of sequencing is came first? Discovered first
Proteins,
time?
protein,
Protein sequencing by scientist
poly,
Sanger, I think.
Sanger or Lynch.
Yeah, Sanger actually determined sequencing the protein discovered first time. Then again, he only discovered DNA sequencing, RNA sequencing. Yeah, that's sequencing. Just RNA sequencing later it came. Then the Frederick Sanger actually is actually the pioneer of this DNA sequencing field or sequencing field. He actually got 2 times Nobel Award. He's such a legendary scientist. He got 2 Nobel Awards, the same area. Not same area, like at least for the sequencing, protein sequencing and DNA sequencing. Okay. When you look in the history of sequencing, it started with 1953 when the DNA structure was deduced by Watson and Crick in 1953. Then after later, when a DNA discovery was made, the scientists were interested to know what exactly the bases present in the DNA and how the order of bases present in the every organisms of every genome, like organism genome. Then in the first time, bacteriophage MS2 was first sequenced by Sanger sequencing method. Actually, not Sanger sequencing, it is chemical based method.
Maximum Gilbert's.
Yeah, chemical based method like degradation method by several chemicals discovered by maximum Gilbert. Then in later 1977, publication of Sanger sequencing method. Then soon after that, 1983, actually it was gel based. You share the DNA, then you add some of the sequences, terminator nucleotides, then you just break the DNA, like you terminate the growing chain and you observe in the gel. But it is so laborious and time taking. But soon after the discovery of PCR, even Sanger sequencing now it is automated. And the PCR actually lets the development of nowadays NGS sequencing. Then in 1986, first automated sequencer by the applied biosystems, like it's called as AB370A. Then after the discovery of automation of Sanger sequencing, later one of the model organisms was sequenced. It is called as C. Cinerability elegans, having 100 base pairs genome. Then after in 2001, beginning of human genome sequencing project, and in 2003, the human genome project was completed and determined there is a 3.3 billion sequence per there, base pairs per there, the human genome. Like, then after that in 2005, parasequencing came, like it's called as Roche 454 sequencing system. Then again Illumina series came. Then now we are at the stage of long read sequencing. It is called as third generation sequencing. We'll discuss. Okay. This is how the DNA sequencing science has developed from 1953 to till now. Still, it's a very competitive field. You read any sequencing method, they just generalize the methods, but they never disclose the information how exactly they are doing, like which chemical they are using, which chemical they are, like which procedure they are using. So nobody actually, only standard methods, there is some protocols were there, but it's highly competitive field. Companies will not even disclose, I mean even Sanger automations, like several steps they are not disclosed. Okay, they got patented but disclosing all, but it is not like it's kept a trade secret kind of things. That's why it's every day we have a new sequencing method. Which one is highly in every lab we are using this sequencing method? The
Illumina,
sequencing platform we are using
Illumina.
Illumina. Okay, because it's a cost effective. It's very cheap. Then when you're looking at what are the different types of sequencing, there are three major classification of sequencing. One is first generation sequencing, next generation sequencing. It is called as NGS. Normally, we many people I observed they write new generation sequence. It's not new; it's actually next generation sequencing. Then third generation sequencing methods. We will discuss one by one. Like I will not go in detail. I will give what to read actually. You don't need everything to be understand by all the sequencing method. You just need how actually it's important for a plant breeder. Like what are the, like whenever we're doing something. For example, you want to do QTL mapping. You want to genotype all your population. Like for example, you have 200 population, 200 mapping population. Then you have to screening with SSR marker. Like at least you need to go 200 SSR marker. It's very very difficult. Then you going for genotyping by sequencing method. Like you genotype all the 200 individual by sequencing, by Illumina sequencing. Then you generate SNPs, and it is highly automated. Like you will get polymorphism in, I think in a 1 month you can get all the data. You just send the DNA to company, they'll give you a sequence data, genotypic data, and you run your analysis. It is very simple. But if you want to run SSR marker, it's very, very laborious. That's why many people, like many laboratories, they are using this NGS sequencing methods to genotype the population. Nowadays, transcriptome is also taking boom. That also many are using Illumina based technologies. Then what are the difference? Why there is a three major classification? Actually, we will discuss this. In first generation, we had two types of sequencing methods. One is maximum and Gilbert sequencing. The another method is Sanger sequencing. And till now, it's a gold standard sequencing method. Even though many methods came up, but till today, it's a gold standard sequencing method. Why? It's considered gold standard sequencing method. Why it is so? Any guess?
It's highly accurate, sir. Maybe
Yeah. In terms of error, you're getting only 0.01% of error. Compared to other sequencing method, error is very, very less. The only problem is it is very costly because it needs a very laborious compared to other methods. The data generated per run it's very, very less in case of Sanger sequencing. But others are in terms of data generation per run, it's very high and very cheap, but having some error. And third generation sequencing, especially, they are famous for having high read length. We will discuss what is read length, what is all the things. Because of high read length, they are famous. Like Pacific Biosec, like single molecule real time sequencing, or Oxford Nanopore technology. We will discuss one by one. Then, what is Sanger sequencing? Every sequencing methods follow one chemistry called as everything is revolved around this chemistry. So DNA replication or a chain adding like a nucleotide adding to the growing chain of the DNA. Every sequencing method it just rely on this single equation, but they use different part. For example, you take Sanger sequence. In Sanger sequencing, it is also called a chain termination method. Chain termination method. Why it is called as chain termination method? We are using a nucleotide like DNTPs. Instead of DNTPs, we are using DDNTPs. Here, instead of adding DNTPs, we are adding DDNTPs. What is the meaning of DDNTPs? Can anyone tell?
dideoxy NTPs.
Yeah, dideoxy DNTPs. Why are we using DDNTPs particularly? Because your DNA polymerase having one unique nature, whenever it having a free 3' OH, then only the DNA polymerase will start synthesize the DNA. It will start adding the DNTPs. But this DDNTPs doesn't have this free 3' OH group. So that your DNA polymerase enzyme unable to add the further DNTPs to the cycle, your DNA growing cycle will be stopped. That is a single principle, a fundamental principles of Sanger sequencing is here instead of while DNA replication, instead of adding DNTPs, we are adding we are adding DDNTPs. What is the nature of this DDNTPs? It lacks free 3' OH. It doesn't have free 3' OH. Whenever the DDNTPs is incorporated into the chain, further addition of DNTPs is actually by the your DNA polymerase. But whichever the DNTPs, it doesn't have free 3' OH. It doesn't have 3' OH. This DNTPs were not added further by DNA polymerase. This is the fundamental principles of your Sanger sequencing method. How it is used and what are the like how the procedure was, like how we actually lay down the procedure, what are the steps. It is like it is not that much necessary for the breeders. Just this chemistry. What is the chemistry of Sanger sequencing? You should able to tell. Can anybody to recall what is the chemistry of Sanger sequencing? Fundamental chemistry.
Forgot,
No,
sir.
not understood or forgot.
No, sir. Forgot, sir. Really forgot. That
Okay,
means
now
it will
only I told.
terminate at particular nucleotide. It will terminate the chain at particular nucleotide. That will make us to determine the last nucleotide.
Yeah, no, no, I'm asking this fundamental principle. You are telling the answer. Whenever you added DDNTPs, okay, dideoxy nucleotide triphosphate, this DDNTPs doesn't have free 3' OH group. Means whenever the DDNTPs will going to add, then definitely there is no free 3' OH. Where DNA polymerase need this 3 OH. If there is no free 3' OH, how DNA polymerase will add further DNTPs? This is the chemistry. Okay. First,
Okay.
you should remember your entire DNA replication path. This is DNA is there, and every DNTP is having free 3' OH. The DNA polymerase will come here. The DNA polymerase will come here and it will add further DNTPs, and you are getting chain extended. Okay. Then whenever the DNTP is added into the DNA, like a growing chain, what will happen? Your nucleotide is incorporated like N plus 1, your N, this is plus 1, DNTP, your DNA plus N plus 1. What is the release? What molecule will going to release? One molecule is
H plus
pyrophosphate,
and
another is
paraphosphate.
N plus. You should remember, okay? It is a fundamental equation of DNA replication. And many times it's asked in the exam how many molecules of H2O will released whenever the phosphodiester bond will make, how many protons will going to release, how many pyrophosphates will going to release. See, it's a triphosphate. Only one of the phosphate will going to incorporate in the DNA chain, but rest other two will release as pyrophosphate. This PPI is called as pyrophosphate. This fundamental equation you should remember. If you remember this equation, all DNA sequencing is very, very easy. Okay?
So
Then,
what about the water molecules, or how many water molecules it will release?
like whenever the DNP, like five water molecule will going to release. Okay? Whenever the DNTP is actually bind 5 water molecules.
Then we can add plus 5H2 on us in that equation itself.
Yeah, yeah. I think I will check. Okay, but whenever you add DNTPs, definitely the water molecule going to release. It's 5H2. I will check once. Either 3H2 or 5H2. I have confusion. I will let you know. Okay, but you should remember all the things. Chemistry of, see, this year they were asked what is the IPAC name of this DNTPs, like 2-oxy, 3-hydra, like even I also forgotten. 2-oxy, 4-aminos, pyridine, 2-oxy, 4-oxy, 2-amino, pyrimidine. Even they asked this IPAC name. Please remember everything about the fundamentals of the DNA. Then this is, you understood Sanger sequencing. Instead of DNTP, in a DNA replication, we are adding DDNTPs so that it doesn't have pre-3' OH. Your further nucleotide extension will stop. Then we can, based on that, we can determine the DNA sequence. How it will be? Please watch any animated video, it will be very clear. Then next, another chemistry we are using for DNA sequencing is pyrosequencing. Whenever the pyrophosphate will release, whenever the pyrophosphate will going to release, this can be detected by, actually whenever pyrophosphate release, it can be converted into ATP. Then further ATP when react with luciferin, it will give oxyluciferin and light. This light can be detected by CCD cameras. Then, for example, you are adding here DATP, adenine triphosphate. Then your pyrophosphate released. Then you can say, okay, this having T. Means whenever the T is there, your adenine will incorporate. Then whenever TTP is thymine is added, but no light is react, no light is came. Like no this react, no pyrophosphate was released and no light is detected. Then there is no uh, complementary nucleotide layer. Like this, you keep on adding the bases, and based on the color expression, sorry, based on the pyrophosphate release, light will emit and it will detect it CCD, detected by CCD cameras. Then we can say, okay, particular base is there in the uh, nucleotide chain. Based on that, we will deduce the sequence. That is called as your pyrosequencing method. I will share some videos about every sequencing method. I have some videos. Okay, I will share that. Based on that, you understand. Okay? It is called as ROSE 546 or 54. Then, in some other companies, they are using whenever the DNTPs is added, there will be release of H plus ion. Means this this is H plus ion is directly related to pH. Whenever H plus added to the solution, then definitely pH will going to increase or decrease. Based on the change in the pH, that can be detected by this one machine called as ion torrent sequencing method. Whenever the DNTP is added into the sequencing chain, there will be release of H plus ion. This H plus ion will make changes in the pH of the solution. Like this can be detected by ion torrent. The system is called as ion torrent sequence. Means based on the H plus ion release, whenever the DNTP is added, H plus ion releases. Based on the detecting how many number of H plus ions are released, we are reducing the which sequence is added. That is called as ion Toronto sequencing method. Then another method, Illumina. Actually, it is also called as, and this ABI solid, both uses same chemistry. They are actually sequencing by synthesis. Means they add pre-3' DNTP, but instead of DNTPs, they are using some fluorescently labeled DNTPs that also act as reversible terminators. Whenever they add, they will emit some color. Based on that color, we can detect. Based on that color, we can detect which base is added into the nucleotide chain. That which is the nucleotide present in the genome, we can able to detect. It is called as sequencing by synthesis. I will share the videos so that you will get one by one. And for SRF, it is not that much important. And even for any exam, it is not that much important. You just know which are the platforms are available and what are the chemistry. You'll see in detail. For every each platform, you should remember mechanism, what is the read length, what is the error rate, how much output data, and which application it is majorly used. You remember if you remember these 4 points for each platform, it is more than enough. Okay. For example, you take Sanger sequencing. The chemistry it is used is dideoxy chain termination method, and read length is 400 to 600 base pairs, and error rate is 0.01%, and it can generate per run 100 kb data, and it can be used for transcriptome assembly, or it can be used for sequencing, either transgene validation, whatever it may be. It have wider application. Then when it comes to 454 sequencing method, it majorly uses pyrophosphate sequencing chemistry. This chemistry, pyrophosphate sequencing chemistry. Then the read length is 700 base pair. The error rate is 0.1%. See the difference. Here 0.1 and here 0.001. That's why it is still regarded as gold standard sequencing method. And it can also resolve haplotypes, or it can be used for DNA sequencing. Then ion torrent. It is based on the detection of H ions. Whenever the DNA, like DNTP is added, then definitely there will be release of H plus ion. Based on detecting of the H plus ion release, this photon will work. Ion torrent of photon. The read length for this ion torrent is 400 base pair, and error rate is quite high than the 454 and Sanger sequence. Error rate 2%. For example, you are sequencing 100 base, then definitely there will be 2 error. This much error percentage will be there. Then it can generate 100 GB data per run, and it can use for splice variant identification, DNA sequencing. These are specific use I am telling. Commonly they are used for DNA sequencing. Just I highlighted which is like, for example, you want to identify splice variants, then definitely you can use ion Toronto, and it is cheaper than that of the other method. 454 and Sanger sequencing, ion Toronto was little bit cheaper. When solid to 4, actually it is having very very less lead. That's why nobody is using solid sequencing, because they are having very very short reads. As where your assembling will be very difficult if you are using this solid techniques. Then it is majorly by the chemistry called ligation of two base coding. Then the error is very less when compared to ion Toronto. It is 0.1%. It can able to generate 100 GB data as same as like ion torrent, and it can be used for gene expression quantification. Then Illumina is most commonly used. It's based on the sequence chemistry called as reversible nucleotide chemistry, and read length is 100 base pairs. Nowadays 150 base pair also you can use. I use 150 base pairs. Then error rate is 2%, and it can generate 1 TB of data. See the difference. Here 100 GB, 100 GB, but here nearly 1000 GB. It is 10 times the solid, 10 times the ion Toronto, and it is 1000 times the previous sequencing methods. That's why many laboratories they are majorly depending on Illumina ISEC. Nowadays, there are several series. Hilum ISEC, TrueSec, HiSec, ISEC 10, ISEC 500, TrueSec 500. There are a lot of series for there. Don't worry about it. Just should know which chemistry it uses. It uses reversible nucleotide method, or it is also called as sequencing by synthesis. Then it majorly used for gene expression quantification like RNA-seq that we will going to discuss tomorrow. Then third generation sequencing. For example, in NGS, what will happen? We'll generate smaller reads. See the read length 50, 100, 400. But when we were sequencing eukaryotic genome, we were getting so much gaps. For example, sorry, we are getting so much gaps. For example, you're having this reference genome, eukaryotic genome. You are getting, when you are assembling, what will happen? We are getting more gaps. There is no reads will going to assemble here. When it is a short reads, it's very, very difficult to assemble. That's why we need to use a sequencing platform which having longer read length. See the read length here, it is 1000 to 10,000. Nowadays, ultra long read sequencing also came, where 100 KB read length also available. But one of the major disadvantages is it is not used like error rate is very very high, 10 to 15%. For example, you are sequencing 100 base pair, then definitely there will be 10 errors. That's why nobody is using. But for sequencing larger genome like wheat or any other biggest genomes, it can be sequenced by third generation sequence. Nowadays what people are using, they are sequencing hybrid sequence. Means you can use both FACBio technology and Illumina technology. The sequencing two times, one by Illumina and another time by PacBio. They're combining both the rates, then they are overcoming the disadvantage of the both the methods and taking the advantage of the both them. That is called as hybrid sequence. I am going very fast because it's not that much needed. If you ahead this table, it will be very useful. And one thing, PacBio generally they are not used for gene expression quantification. Why? Because whenever you're having long read length, what happens whenever you're taking the count per exam, it will be very very difficult. When you're having short reads, you can easily take the counts per exam. Then where can you generate easily the expression count matrix? It will be used for your expression analysis. But in PacBio, what will happen when you have long reads, like you can't count how many reads per exam. It's the main basis for your RNA-seq. That's why it is not generally used for RNA-seq, but rest are used for gene expression quantification. I hope like at least you got some points. I know it is not clearly you will not get at a single time. I will share the videos, you watch the entire chemistry of e-sequencing method so that you will get some idea. Okay. Then we will see how the general workflow of all these NGS. Okay. First, what you have to do, you have to take the DNA sample, for example, leaf or whatever it may be. You isolate the DNA. After isolating DNA, what you have to make? You have to prepare the library. First, your DNA is very long. You should make fragmentation. You should make smaller fragments so that it will become fragments. Then there are different methods to share this DNA. One is mechanical shearing, another is enzymatic cleavage, or another method is your bead-based technology. Bead-based technology. Nowadays, most probably used technologies to fragment the DNA is bead-based technology. Then, after that, you just fragment your DNA. Then you had to ligate with adapter. You know what is adapter? Adapter is a oligonucleotide sequence of known sequence. For this adapter, what will make? There are two types: single-end libraries and paired-end libraries. Either you can use both of these. Means, for example, this is a fragment. Actually, I don't have pen tab. It's very difficult. For example, you are adding adapter and you are reading of adapter from only one direction. From the one side of your DNA fragment, it is called a single end. But you are ligating adapter from both the sides and you are reading in the both the direction. It is called as paired end. Whenever you are making adapter, when you are amplifying, then they are called as reads. Means at a time you are giving a single read for a flow cell so that it will going for sequence. You understood what is fragment, like how to shear the DNA either through mechanical injury or through bead-based technique or through enzymatic cleavage. Okay. After cleaving, your short fragments will come, and each fragment you add adapter of known sequence. Then this can be used for your amplification. Like either there are two types of amplification: bridge amplification or emulsion Fisher. Don't worry about that. Nobody will going to ask you. Then after you have to amplify each fragment into millions of copies. For example, one fragment you should at least millions or 10 to the power of 5 times you should generate multiply the each fragments in a very large number of. Then after ending of your library preparation you have to go for sequencing. That we will discuss next slide. Then you can generate your bioinformatic pipeline. We're going to discuss in detail. Then what is flow cell? Each
Sir,
illuminant. Yeah.
sir, do we sequence all the millions of clones what
Yeah,
we prepared
everything is sequenced. Every fragment is going for sequencing. Okay.
Okay, sir.
That's why that is called as depth and coverage. I will come to that. Okay. We
Okay,
had to, because now what will happen? Why we need to go amplification? See, for example, Illumina, I told you are using some fluorescently labeled nucleotides called as reversible terminators. Whenever it added, it will emit the color. For example, your one DNA, for example, if single fragment is there, only single time your DNA will going to add and you have to reduce the sequence. But the color detected from one fragment is very, very having low intensity, having very low intensity. It cannot be detected by your CCD cameras. Means whenever you are adding this fluorescently labeled DNTPs for your reducing the sequence of the read, the color releases is the intensity of the color was very very less, so that whenever the low intensity cannot be detected by the cameras. What can we do? We should increase the number of reads of the same fragment so that the color color released, the intensity will become very high. When you are having millions of fragments, from each fragment your color will going to emit, then intensity will going to increase, that your CCD cameras will able to detect. That is the main reason why you are going for amplification. There is another reason to reduce the error. Means, for example, there is a one terminology is called a sequencing depth. Means, for example, here A is there. How many times this particular A is sequenced? For example, you are sequencing the A, same A with 10 times, it is called as 10x. You are sequencing same A for 20 times, it is called as 20x. You are sequencing same A for 30 times, it is called as 30x. Means how many number of times a single base is sequenced. That is called as your depth. That's why whenever you are listening about the sequencing data, they will ask you how much depth, okay, then how much coverage. Coverage I will come. For example, you are having 450 MB genome. Coverage means how many 50x, 50 times you have to sequence whole genome. That is called as coverage. When depth is each base is how much time, how many times each base is sequenced. That is called as depth. Okay, that is a major. How why you are having higher depth will be higher advantage because error will be less. Where 50 times you are confirming here A only there. Where 10 times, for example, you are using 10x, 10 times you are confirming that oh here A only there. 50x means 50 times you are confirming that your A only is there. Like that, if you are more depth, you are going to get a very good accurate data with very less error. And higher coverage also will get less error and data will be very good. That is easy for assembly. Okay, understood?
Yes, sir.
You understood what is fragment, what is read, what is depth, what is coverage. It's very, very important.
Yes,
Then,
sir.
then it's a flow cell. Means for each line we are
Sir,
loading each read. Okay, yeah.
sir, can you explain what if the read is less than the fragment of the DNA? And what if the read is larger than the fragment of DNA? What will happen
Yeah,
inside that
that
time?
will come. Actually, whenever I will tell that one, well, alignment, like mate pair rates, okay, that gaps, I will discuss. Okay? In alignment, I will discuss them. If you tell now,
Okay,
everything will be spoiled. Okay, what is that? While alignment, it will be a
okay,
problem. Like how much, if you have a reads like more than the fragment size, what will happen? If the less the it's called as mate pair distance, that will cover like gaps, mate pair distance. In alignment, I will cover this. Okay, if I tell everything
okay.
will be spoiled. Then this is the flow cell, which you are adding all the reads here where exactly sequencing will happen. Like this, so many billions of flow cell will be there. So that's why you are generating 1 TB data per run, 2 TB data per run because of this discovery of the flow cells. Then how actually it looks inside the flow cell. First, you're having the fragments. You're adding an adapter for each fragment. Then there will be flow cell. This is called as flow cell, where each adapter will go and bind. Where then what happens? They're having primer. They will bend like this. They will attach. Then next to that there will be another this one. They will the while DNA polymerase will actually amplifies. This is called as bridge amplification. Then after that, each can be used, this sequence can be used dissociated and where whenever the dissociation, this emitted and this can be regarded this A, T, G, C based on the color, we can deduce like based on the color signal, we can deduce, okay, a blue color release, okay, T will be there, A will be there. What is the like yellow color is there, A will, orange color is there, A is there, green color is there, G is there. Like this one magenta color is there, C will be there. Like this, where reducing based on the color signaling. Actually, now by picture you will not get anything. While you are watching the video, then only you will get. Okay, like I will share that videos. Don't worry. Then what is the workflow? See, platforms. No,
Yeah.
even you remember or not remember, nobody will ask you actually. Just give company, they will give raw rates. Up to here they will give. But after that you should know these things actually. Today I am going to deal with these things today. Okay. First, after doing all those things by different platforms, we will generate a raw reads. Either it may in FASTA format or FASTQ format, where fast queue means, remember, raw sequence read in fast queue means it's having a quality check. Like sorry, per base the quality score is uh, assembled. For example, how to check the quality I will show. For each base, whenever you're sequencing, for each base, we are having a quality score, like Fred score. Normally, above Fred score 30 only, we have to choose for further downstream analysis. What is that means? For example, you are sequencing 100 base pairs, like minus log 10 to the power of p. Minus log 10 to the power of p, where p is probability of occurrence of an error. For example, you are out of 1000, out of 100, sorry, out of 10, you are making one error. Means log p, one is like log 10 to the power of 1 by 10. You will almost get where 10 is the quality score. Then for 100 base pair, actually writing is very difficult. Log 10, this is p value, probability of occurrence of error. If we substitute this value in this log 10, you will get this Fred score value. Means how many times you are conducting error. For example, you are sequencing 10 base pair, you are making one error, means the quality score is 10. You are sequencing 100 base and you are having one error, means quality score is 20. You are sequencing 1000 base pair, but you are getting only one error, means quality score is 30. The formula is log 10 to the log 10 into bracket p, where p is your probability of occurrence of error. Like you just remember a simple trick: 1 0 is there 10, 2 0 is there 20, 3 0 is 30. Definitely there are many times asked in the exam. This is called as Fred score 10, 20, 30. Means if you are making one error in 1000, it will be good. But you are choosing one error in 10. See how many million base pairs you have to sequence. For 10 base you are getting error, your all sequencing will be spoiled. One in 100 also it will be very costly. Like error will be very costly. But you are making one error in 1000, it will be at least good. And more than that, it's not maximum. It is a minimum error. Okay. If you have Fred score 40, it's also possible. Fred score 50 also possible. Okay, whenever higher depth, definitely you are going to get less error. That's why when you remember, we are making one error in sequencing 10 base pair. We are making one error, means Fred score value is 10. We are sequencing 100 base pair, but one error in 100 base pair is Fred score value is 20. We are sequencing 1000 base pairs, but one error in 1000 base pair, Fred score value is 30. This should be checked before completing your sequencing. Okay. Understood what is called Fred score value? Any doubts regarding Fred score?
Yes.
No,
Sir,
not
that
understood.
fast
Not
queue
understood.
means, sir? Understood, sir, understood. But what is that fast queue means?
Sequencing files. See, after sequencing, the sequencing information stored in the FASTQ format. I will show.
Okay, sir.
Okay, first I will explain, then I will show step by step. First, you're having a raw data, it's a FASTQ file. Then you have to do quality check, like above 30 only you have to keep. Which bases which having below 30, you just remove that. Then you have going for alignment. There are two types of alignment. Either you can go for de novo assembly or reference based assembly. Then you have to do variant calling, like alignment means you are mapping the reads to the reference genome. If you don't have a reference genome, you just go for de novo assembly. But for time being, we'll just discuss only reference genome assembly. Then after alignment, I had to go for variant calling. For example, here A is there, which is the if A is there, okay. If any other base is there, that can be regarded as variant. Then based on variant calling, we can generate VCF file. Then you can do annotation, functional annotation, structural annotation. Then you go for database. Okay. Then this is what a general protocol. First, you're having a raw data, which format first queue files. Then you have to do quality check based on Fred score. Then you have to do alignment. Then you have to do processing. Then you have to do variant calling. Then you have to go for annotation of that variant. This is how the NGS workflow will actually happen. Then these are the different sequencing machines that used for either Illumina, either different solid, solid, ISEC 200, GSFLX. These are forms of Illumina. Every day there will be new sequencing platform will coming up with a more efficiency.
Sir,
Yeah.
sir, that variant call format means that particular base will be different from reference genome.
Yeah. Whenever A is there, but at the same position, you may get different nucleotide that can be regarded as your variant. We will show one by
Uh,
one. Wait, we'll see. See, actually, it will be in zipped format. We need to extract. Time being, test files. Seeing my screen?
Yes.
yes, sir.
Yeah, this is actually FASTQ format. And normally, every time, why this is see, you're seeing first row. Actually, every FASTQ, every read having 4 lines. Remember, FASTQ format means every read having, for example, when time your read is sequenced, every read having 4 lines. That is called as FASTQ format. For example, at first line is identifier, means for example, whenever doing bridge amplification, whenever you do bridge amplification, here there will be one site. Here you are seeing like for example, red color one spot is there. Like on a flow cell, there will be a spot. For example, this fragment is sequenced 100 million times. But how you are going to know that from here only that fragment is there? It is called as an identifier. Like it is called as an ID number. Based on that, we can sequence. Okay, this read is belongs to here only. That's why we are having identifier at ERR, HAW. This address. Take this. You are giving address for each read. See, millions of reads were there. Like for example, you take 450 MB rice genome and into 30. 450 million base pair into 30. How many million base pairs? Divided by 150. 150. Like read length is 150. Nearly how many? 30 million reads if you are generating means. How you locate that all 30 million reads? Very difficult. For each read, we are giving an address. Okay, here from where the read is coming, that is called as identifier line. Means you are giving an address for each read. Then the second read is your nucleated sequence. C A A G G. You are seeing my screen or not?
Yes, sir. Yes, sir. It's visible.
This is sequence. Then there will be plus symbol, means joining next. Then this is CCF. You are seeing this one. This is actually quality score. Actually, if it is in letters, for example, if it is in A, B, C, D, your quality is very high. But if it is in punctuation, like star mark, hyphen mark, or any comma mark, it means that quality score is very less. Actually, in manual, if you see the quality check manual, you'll get all those things. But as of you now remember, FASTQ file means there will be each read having 4 lines. One is identifier, one is sequence, one is adder, like plus, another one is quality check. For each read, there will be Fred score. Normally, if it is ABCD, if it is in a letters, then normally we regard as quality is very, very high. Okay, understood what is first key file? We'll
Yes,
take
sir.
this one. We'll save this one. I think it's a human genome. So big. Then, how do you check? You're seeing my terminal.
Yes, sir.
Wait, actually, it will take time. Either you can download any small sequence, you can do quality check and all this thing. Fine. First, you have to go for directory. Then, I mean, still it will take time. For example, this is the command line. You have to first see. I have already generated QC report. It will take a lot of time, okay? But I will just where is yeah. See that sequence we have extracted. That one will run. Any sequence you take. Yes, Wanda. It is installing first QC. Like I'm using different environment. That's the problem. In Panda, I was using RNA-seq environment. Actually, whenever you're doing all this bioinformatics analysis, you should learn Linux operating system. Okay, there you can access Ubuntu like Anaconda. It is a distributor of Python. Yeah, started. Now fast QC. I'm running fast QC. I hope it will be completed. See how I made it. Or observing. See, these are
Yes,
the
sir.
things actually. First, you should go for Linux command like Linux is to install Ubuntu. It is either Linux can be in two ways. Either you can install completely through your, for your laptop, or you can along with Windows you can use Linux. It's just like a window. Or Windows operating system. Linux is also one of the operating system. Then what you have to do after installing Linux, like Ubuntu is one of the distributory for Linux. After installing Linux, what you have to do? You have to install. First, you have to generate. You have to learn how to use these codes. For example, ls is for listing the directories. Then, like I was in home directory first. Then you have to go for your cdd, like just like it's Windows is having very easy graphical. See, like here you having graphical interface. Wherever you want to go, files you are going. But here you can't use graphical. You can use, but command interface is very easy. Within like don't need much time. You just based on the commands, you can go for multiple directory. Then it's just like you install packages, then run your this one. The analysis completed for this one. We'll go for
Sir, which is better, software or Linux?
yeah.
Sir, for analysis, which is better, R software or this Linux, sir?
No. R software is good, but in bioinformatics, like most of these sequencing based, NGS based packages were built in Linux operating system. Like they support Python. If you learn Python, you don't need this also. It's just an operating system. You can run anything. You can run R here. It's not this one. You can run R here. You can run Python. It's just an operating system. How Windows is there now? It is Linux is operating system, but it is just like you're handling the system on laptop only. You can use R here. You can use anything here. Okay, it is not like that Linux or R. It is just operating system. Here it is very easy. Many packages are built in this one. If you are using same packages for Windows, it's very costly. Like no, I think nothing is there. I think it's completed. I hope it's complete. It's still running. Like first you have to run the fast QC. I will show how fast QC files will be there. What you are going to get from fast QC. Sure. See, fast QC will be, it is multi QC. Like for one sample I did, you can look at multiple samples at a time. Okay, that is called as multi QC. Where normally it is looks for how many number of counts you have. Sorry. How many number of files we have, like sorry, how many number of sequence you having. Then you can look for quality score, then per base n content. And please read this assignment, this is take as an assignment. Read fast QC manual so that you will get what are the things were there. Then what are the percentage of duplicate rates. See the quality histogram. See, all are above 35. That's why it means that your quality of the your sequencing data is very good high. Then you can see the adapter content, that is also very important. After analysis, you have to remove all your adapter because you are adding adapter. It is not originally present in your sequence. You have to remove all the sequence or adapter sequence. Like adapter content is almost a zero, I think. Then you have to check all these parameters: n base content, GC content, duplicate rates, red scores. After that, you decide either your sequencing data is good or not. Then you go for downstream analysis. I will check whether it is or not. It will take a lot of time actually. We're having a very small sequence, it will be very easy to do. One day we will sit. Yeah, see, you are seeing this one, this QC
Yes, sir.
this past QC report is generated. See,
Yes, sir.
basic statistics, per base sequence content, per sequence quality scores. See, average quality score, it is nearly 30 to 40. It means your data is very good. Means it should be above 30. Then per base n content, see, per base sequence content. See, this graph is so, means here per base sequence content is not that much good. Then per base n content, it's almost an basal level, it's very good. Then GC content, it's near to the mean, and red line and blue line should overlap. It is very good. Then there are several. If you read the manual, party check manual, you will going to get. And how much over a present sequence. What is adapter content? This is very, say adapter content nearly zero. Means you are sequencing. Please you have to remove the adapter. For removing adapter, there is another package called as trigonometric. I will also run that one also. Yeah, I think it is not installed. I have to install again. But I have to see the code. Now, another thing, remembering code is very difficult. That always you have to make one folder. Like this is, like for removing adapter, you have to use trigonometric. Okay? Like it's a very lengthy code. I'm not going to use. It takes a lot of data. Okay, just you're removing adapter, you have to use trigonometric. For fast QC, fast QC, quality check, fast QC. For alignment, BWA or fast, like I use several. See what analysis, why I'm going to run actually. I will show the history. Yeah, Here I am running I run trigonometric actually yesterday. Trigonometric, I have to mention which spread score 33. Means I am taking only the sequence which above 33. Then you have to mention all your sequence information. Then it will like adapter content, everything you have to mention. Then a read will be read. Then after that you have to like for alignment also had it. So for alignment there will be a code called as either you can use any of the software for alignment. Like I am using HiSat here, like HiSat alignment tool. Either you can use any tool for this. Okay. This is about sequencing. If you have any doubts, you can contact me. After running, after alignment, you will get like alignment. I can't able to do because it takes a lot of time. For rise, at least it take nearly a whole night. Okay. It's very difficult to show here. For small genome, I will take any sample. One day I will sit. One day we will completely will discuss how to do this everything. So then after do data after that you have to do data processing. Then variant annotation. There are separate commands for that. Okay, everything will have. It's like one code. If you run that code, everything will come. It's not like a magic. Okay, everything you can also learn. Okay. Any doubts up to here?
Sir,
Any doubts up to here?
it is so complex that
No, no,
running
no.
Linux and
No. See, in MSc I did transcriptomics. See, NGS leave. I have run transcriptomics. That is too difficult. I have to learn. Okay. Is world is so competitive. You should learn many things. Okay. Bye bye. Bye. Any
okay.
doubts you can ask? Okay. Tomorrow we will discuss some other RNA-seq. We will discuss after RNA-seq. We will go for your like reverse analytical this one. Then we will end up the class. Okay. Bye. Good night.
Sir,
Yeah.
sir, for bioinformatics aspects, are there any reference materials to study?
No, I just read it from YouTube. Okay. There is a one bioinformatic coach YouTube channel is there. I learned everything from there only and my senior. My
Sir
senior
means,
was there.
from SRF point of view,
No need to
last
worry all those things. Okay. This will not come for SRF. Okay. Those who are doing in those who are doing mapping studies, for them I just discussed. It is not a magic. You can also do. I'm just showed how we can do. Okay. You can able to do yourself jivas, you can able to do a qtl mapping if you are do yourself, you have to do yourself transcriptomics. Okay, analysis you should learn, should be learning by yourself, then only you will get like you will get good reputation. Okay, anything.
Sir, what is the key difference between this NGS and the third generation sequencing?
Yeah, I not discussed the read length. Here we having maximum read length. See in NGS what is the read length? And oh, okay,
700.
sorry I forgotten. Actually, what is the main difference between NGS and third generation sequencing? In NGS, for each read, we are amplifying. But in third generation, we are not going to amplify. It is called a single molecule sequencing. Every read is directly go for sequencing. That is one of the advantage. Okay, like there is no need of amplification in third generation. In NGS, we need, this is the main difference. Here, we are not amplifying fragments. Okay, but in NGS we are making millions of fragments of each copies by using bridge amplification or emulsion amplification. But in case of third generation sequencing technique, we are not amplifying anything. That is called as single molecule real-time technology, SMRT. We are using directly single molecules for sequencing. That was that actually in PCR, while doing PCR, for PCR thermus aquaticus is very error prone. It may introduce some errors while doing PCR. That also generates high error. But in case of this one, you may your amplification bias is reduced. But you are getting because of long read, you may get error because of other reasons. Okay, that is the only main difference. We are not amplifying the fragments in third generation sequencing, but we are amplifying by bridge amplification and this emulsion amplification in NGS, but in third generation sequencing we are not used. That is the main difference. And read length. Here Illumina max to max is 100 to 150, or max to max you take 400 in 400 to 500. But in case of NGS, we have in third generation sequencing 1000 base pair, 10,000 base pair. Read length is very very high, so that whenever you are sequencing eukaryotic genome, you are getting less, less gaps. In RNA sequencing, I will discuss like what asked mate pair distance, fragment size, that everything I will discuss in RNA sequencing. They have
Sir,
complete chart. Okay.
sir, single molecular sequencing in the sense without amplification, we'll sequence whatever the DNA you
Yeah,
extracted.
yeah, 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 yeah. That is the idea.
Okay, sir.
You are fragmenting, and this fragment directly go for sequencing platform. Then there are two techniques, nanopore and FacBio. I think I will discuss. I will share the videos so that you will get what is nanopore, what is Oxford, what is nanopore and what is FacBio. We're going to get. I will share those videos. Okay? Don't worry.
Okay, sir.
This third generation also use the same chemistry, sir, like NGS. That
Yeah, same
equation
chemistry
only.
only. See, like in nanopore, we have using different chemistry. Like there is a nanopore. Whenever DNA go into that pore, there will be change in the electric current. There will be change in the some electric current in the membrane. Actually, nanopore is a pore in a membrane. Whenever your DNA pass to that membrane, like in that pore, there will be change in the electric currents in the membrane. Based on the change in the electric current, we detecting the which sequence is added. That is called as nanopore. But in PacBio, there is a different technology. Like there is a one diode will be there. Whenever your DNA will pass through that diode, actually while DNA replication, it actually uses DNA replication mechanism. This small diodes will be there. It will actually detect the currents. Like I will show tomorrow. I will discuss. If you, I will share the videos. First, you watch the videos. Tomorrow, I will. If you are again, you are not getting, you can ask tomorrow. We will discuss it. Okay?
Okay, sir.
With picture, it will be very nice. Okay. Bye. Good night.
Good night.
Bye, sir. Good night.
Good night.